We have been trying to understand yet another a very extremely important endocrine gland, and what did we identify it as? Cortex, adrenal cortex. You remember we talked about adrenal cortex, and then we focused on an important hormone, that is aldosterone, and we classified it as what kind of hormone was it? It is a it's a mineralocorticoid, and its main function was to conserve, okay, to conserve what? Sodium, okay, and also to get rid of the potassium ions. That was, and what was the main dominant factor that regulates the release of aldosterone? Angiotensin 2. You are right. What was it? Angiotensin 2, and for that again we go back to the kidney and that entire system of, okay, we have done that. Okay, now we will go to the yet another equally important, okay, second hormone by the name which is, we will take it as an example, that is a glucocorticoid. Now please remember, these two words are used interchangeably. One is corticosteroid or corticoid, same thing, okay. I mean that word has come so often that people do not even just say corticoid or glucocorticoid or mineralocorticoid or corticosteroid, whatever. In chemistry, they are steroid hormones, and so we will talk about this interesting. So this is being secreted by, you remember there were 3 layers, okay, glomerulosa, okay, outermost, okay, fasciculata. So the fasciculata, the cells of the fasciculata, those cells which are arranged in a radial strands of cells, those cells are a source for cortisol. So cortisol comes from where? The zona fasciculata, which is the second layer in the adrenal cortex, is the main source of this hormone, which we call as cortisol. Great. Okay, and what is the peculiarity of this hormone? Well, it's a glucocorticoid. It's in long term it increases the blood glucose level, okay, it acts on the, but one of the very characteristic features of this hormone is that it's a great promoter of gluconeogenesis. Means what? Formation of glucose from non-carbohydrate source, which means fats and what? Fats, yeah, fats and proteins and fats. Okay. So let us see. If you take a slightly global view, you will find that what does insulin do? Okay, suddenly a lot of glucose in the blood after food intake. Okay, then you conserve it because there's plenty of is there. Okay, how do you conserve it? Well, you first take it to the liver, convert it into glycogen. You take it to the adipose tissue, convert it into fats, and store. I mean, you are in an energy rich condition. Okay, and then as the time passes and now you are starving for a while, okay, you haven't had food for another 2 hours, then the glucagon comes into role. You have done glucagon also, and glucagon will do something opposite. Okay, so whatever has been stored has to be, but if you starve for a longer time, okay, now starvation for a longer time is almost unheard of in since our civilization, but for an animal in the wild, starvation for a very long time is a very common phenomenon. Okay, I mean, you see, if a carnivore like a tiger or a panther has had good food today, what is the guarantee that it will get next? And there is easily a period of 7 or 10 days when the food is just not available. Okay, and there's absolutely, I mean, till again there is feast. There is a feast. So fasting and feasting. So during that period, you can't completely depend on the stored carbohydrates either in the form of glycogen or in the form of adipose tissues. Under those conditions, utilizing the sources from the non-carbohydrate sources means what? Proteins. And again, when I mean by proteins, what do you mean by skeletal proteins? Okay, that's a great source. I mean, it's an expensive source, but it's a great source because the animal has already invested a lot in making of the molecules, the actin, myosin, and a range of other proteins that bring about the mechanical part, the muscle part. But that is a great source when you have been fasting for a very long time, and that can be mobilized by cortisol. So what does cortisol do? It will mobilize proteins which are stored in the form of muscles, number 1. Number 2, the long-term storage in the form of fats, adipose tissue, okay, the triglycerides, okay, you break them down to fatty acids and glycerol, and then use it as energy. So we call it. So this is a main hormone, which helps us to cope up with a starvation for a very long time by continuously look. As long as life is there, for a human being, blood sugar level cannot go below 80 mg per 100 ml. Period. Got the message? Got the message loud and that's it. You can't. You are a human being. You have to, as long as 80. That is the limit. Okay. And you don't get food for 7 days and 10 days. Okay. Then what do? You keep on digesting your muscles under the influence of cortisol. What is the aim? You take those amino acids. Okay, break down the muscles and to amino acids, take those amino acids to the liver. In the liver, the amino acids will convert them into glucose, send them into the blood, make sure that the blood sugar level remains at 80 mg per 100 ml. Got the message loud and clear? So whether you eat or you don't eat, okay, you are now actually eating your own muscles. Who is doing it? The cortisol is doing it. How? Digest the muscles into amino acids, take them to the liver, deaminate them, convert them into a glucose molecule. One aim, because you can spare all your muscles, but you cannot spare your brain by reducing the level of blood glucose supply to the brain, which has to be at the level of how much? 80 what? Mg what? Per 100 ml. That is the level. I mean, you see, as far as brain is concerned, the amount of oxygen that is there in the blood and the amount of glucose that is in the blood is non-negotiable. It has to be so much. Okay, and for that you can draw from anywhere in the body you can draw. So let us look at this very interesting molecule which we call as cortisol. Okay, we have done this yesterday, but yesterday our hero was aldosterone. Today our hero is different. It is cortisol, but the story is more or less same. What are we doing it? So we have the cortex there. From the cortex, now I'll take a cell. Now this cell is from the zona fasciculata, one single cell, and then I have this single cell plasma membrane of the cell, and then as we saw yesterday, the LDL. What does LDL stand for? Low density lipoprotein, and it comes along with the blood and it is internalized by the plasma membrane by a receptor mechanism. So there's a protein there. There's a protein there in the plasma membrane of all the steroid secreting cells. All steroid secreting cells will have that protein. Whether you are in the adrenal cortex or whether you are in the ovaries or you are in the testes, they are all steroid. If I just ask you, tell me 3 different sources, steroid secreting sources, you will of course name what. Adrenal cortex number 1, and then testes in the male and ovaries in the case of female. These are the main sources for the secretion of steroid hormones. In the plasma membrane, you will have a receptor. That receptor will mediate the influx of it. Just doesn't flow. Okay, it's mediated. Then comes and then the cholesterol comes in. You know, the cholesterol is a molecule which is extremely labile. It can actually go anywhere. But if the cell wants to store it for future use, how do you store it? The answer is there. You will convert cholesterol into what? Into an ester. Okay, the cell will do it ester, and then it can be made into a vesicle, and then it can stay. Yeah.
Oh. It is a lipoprotein, right? So it is basically covered in lipid. Why does it need a receptor to get internalized?
Which one?
LDL.
You mean this one? Oh, it's a very large system. It's a large system. No, it can't go. It can't go. It's a lipoprotein system. Okay, so it's not just a lipid molecule that can go. It's a much larger system. Okay, now then so it goes in, and then here is an interesting connotation that author wants to give, that you can store the cholesterol in the form of what? Cholesterol ester. Okay, and then when necessary it can be released, and then when it can be released, then comes a very interesting molecule that we have already spoken about it. There's a protein that is sitting on the mitochondria called as what? Star. What is the full form? Steroid acute regulatory. Okay, so this star will take it in the mitochondria. In the mitochondria, it will be acted upon by what enzyme? Same enzyme. What is it? Cytochrome. Cytochrome C 450 SCC. Side chain cleavage. So that cholesterol is converted into a smaller molecule, pregnenolone. So again, same. We are just Just repeating yesterday's story. Pregnenolone, then it goes into the endoplasmic reticulum, and then it again goes back, and then the cortisol is finally synthesized at the level of the mitochondria, and then the cortisol is ready for the ready for the release. And this is all under the control of the, I'll talk about it again later, it's again under the control of the hormone ACTH. What did I say? Say that again. ACTH. Say that again.
ACTH.
Adrenocorticotrophic hormone. What did I say? Adreno. You just have to know it. Corticotrophic hormone. Okay. Its adrenal is adrenal. Cortico is adrenocortex. Trophic is to grow. Trophic is what? To grow or to promote the growth. Adrenocorticotrophic hormone. That hormone is coming from where? Pituitary gland. Which part of the pituitary gland? Anterior or posterior? Anterior pituitary. So, anterior pituitary gland, okay. Tell me the name of the anterior pituitary gland hormone that we have studied in some details.
TSH.
Growth hormone, number 1. What did we do? And then second TSH. Number 1 was what? Growth hormone. And number 2 was what?
TSH.
And the third one I am talking about is now what?
ACTH.
ACTH. Okay, got it, got it. This is the third hormone which comes from anterior pituitary gland, ACTH, and this and all these biochemical steps are being all the enzymes involved in this will be upregulated under ACTH. We'll talk about it ACTH a little later. So yeah.
Where is the lipid droplet stored? Like, because it's
You mean here? You mean?
once it's converted into ester.
Okay, okay. Oh, it's in vacuoles, endoplasmic reticulum. And separate compartments made of endoplasmic bilipid of layer. Vacuole, some vacuole will store it as, and in the ester form it can be stored for quite long time. And then when ACTH comes, as and when required, when ACTH comes, okay, then those enzymes will convert as cholesterol ester back into cholesterol, then it can diffuse out and then it can. Okay, so here we have the cortisol. Okay. So we are looking at the cortisol and it acts on its receptor. Now, you can ask a simple question, on what kind of cells cortisol acts? I have already told you, it acts on all skeletal muscles, it acts on all adipose tissues, it has abundant action on the brain, everywhere. Cortisol has profound action all over. I mean, it's difficult to say that this particular place is, smooth muscles have relatively low, but it's almost all other tissues are under immune system, lymphocytes, huge number of receptors for cortisol. What am I talking? We are discussing as to what are the sites in the body where cholesterol is likely to exert its effect. What is the answer? Profound effect on liver, profound effect on all skeletal muscles, profound effect on entire immune system, profound effect on the adipose tissue. Some of the main organs on which the cholesterol will have the effect. Now, how does it work? Okay, so this is a cortisol steroid molecule, no problem. Just go inside the plasma membrane. Once it is in the cytoplasm, it is coming across its receptor. Okay, what does GR stand for? Yesterday MR stood for what? So today GR will stand for what? Glucocorticoid receptor. So there are two proteins. Okay, it's a dimer. Okay, they are not a dimer yet. Two separate proteins, identical proteins. When the cortisol combines with these two, okay, but these are attached to a chaperone protein called as HSP. What does HSP stand for? Everybody should know this word. HSP word will come very often. HSP stands for what? Heat shock protein. HSP 30, HSP 50, HSP 50. There is a whole family of proteins. And the receptor is bound to the HSP molecule and it is inactive the form. When the cholesterol comes, cholesterol will combine and then HSP will move away, and then now the two protein molecules will come together and you have a dimer. Now, once it is dimerized, then it can enter into the nucleus. It will find out its suitable response element, DNA, whatever it is, and then it will give rise to this is glucocorticoid response element, the segment of DNA molecule where it will combine, and it will give rise to mRNA, and then you will have the response. What do I mean the response? Let us presume that this is a skeletal muscle cell. Okay, and then you have been starving, no food for 24 hours, 48 hours, 36 hours. In that particular scenario, then this mRNA will give rise to huge number of proteins which will start breaking down the large protein molecules in the skeletal muscle cell. Did you get the message? Start breaking down and converting those proteins into what? Amino acids. Okay, and we have already seen those amino acids will go into the blood. From the blood, where will they go? into the liver. In the liver, they will be deaminated and the amino acids are converted into carbohydrates, and this process we will call as what? Gluconeogenesis. Gluconeogenesis. Okay. And what are the this we have already spoken. Let's see. So cortisol can act on the muscles. Look at the top arrow. Protein degradation will go up, protein synthesis will go down. Okay. So in a cell, so on say as if the entire enzyme machinery will shift in such a way that the proteins will be broken down, glucose utilization goes down, sensitivity to insulin goes down, and then the amino acids will be taken to the liver. From the liver will give rise to glucose, and it will cortisol will also act on what is this tissue, please? Adipose tissue. This will have huge flat fat globules. It will be converted into glycerol. Glycerol will go, and again glycerol will also be converted into glucose, and you will have the homeostasis with reference to glucose in the this is more specifically the fat adipose tissue fat under the influence of cortisol, fatty acids, glucose, amino acids, proteins being broken down into amino acids and being brought into circulation. Okay. Now, whereas this is interesting, whereas the cortisol has profound influence on all the tissues. Now, there is an exception is coming. What is the effect? What is the effect? Bring down the breakdown of proteins, trigger all the enzymes, and we took a classical example of a skeletal muscle tissue. Okay, so far. Similar effect on adipose tissue, on immune system. The exception to the rule is liver. Means what? Does cortisol bring about the digestion of proteins in the liver? No. Then what does it do? It actually promotes the synthesis of a large number of new enzymes and proteins in the liver cells, which play a critical role in deaminating, in converting the amino acids into carbohydrates and fats into carbohydrates. So, the protein concentration in the liver cell actually goes up. So, you can exactly divide the action of cortisol into two categories. One is extra-hepatic. What do you mean by extra-hepatic? Everywhere other than liver. And what that is what we have seen just now. What is extra-hepatic? Break down the proteins, convert them into amino acids, and in the liver it is exactly opposite. Okay, what do you do with the amino acids? Of course, you convert them into carbohydrates, but then you have far more number of proteins synthesized. What are they? Because they are the enzymes. You need them for doing the function of the liver. And what is the function of the liver? Gluconeogenesis, which means convert the non carbohydrates into carbohydrates. So we need to clearly divide the action of the effect of cholesterol into these two categories. Okay. Same with reference to the effect of cortisol on the fats. Increases the concentration of free fatty acids in the plasma, which also increases utilization of energy. Cortisol also seems to have different effect. Okay. We know very well that in the case of prematurely born babies, one of the major problems is the occurrence of or the absence of what? Surfactants in the lungs. As a result of that, the lungs have a problem in the alveoli have a problem in fully opening because of the surface tension. You remember what I talked about? Okay. Cortisol being made available to the fetus at the stage of about 25, 26, 27 weeks of gestation is responsible for the formation and release of secretion, that is surfactant, from the type 2 alveolar cell. Means for the type 2 alveolar cells to become fully functional in the lungs, okay, in the lungs, okay. Who plays a key role? Cortisol. Cortisol plays a key role. Do you get the importance? So in the development, this is a let us try to understand what I have highlighted in the blue font. Can somebody interpret for me? Mice with homozygous disruptions in corticotropin-releasing hormone. Now let me explain to you what is corticotropin-releasing hormone. ACTH comes from where? Very good. Comes from where? Very good. ACTH comes from anterior pituitary gland. Good. ACTH itself is under the control of a hormone from the hypothalamus, because we have already seen that pituitary is under the control of a triggering factor from the hypothalamus. That hypothalamic factor is called as what is it called as loudly? Corticotropin-releasing factor. Say that again. Corticotropin-releasing factor. Now, what is this corticotropin-releasing factor? It is again a small peptide. It is a 41 amino acid peptide that is being synthesized by certain neurons. Okay, so far, as the name indicates, corticotropin-releasing peptide or a factor, it comes from certain neurons. Now, can you tell me the way it travels? It is released by the neurons at the level of median eminence. At the level of median eminence. In the median eminence, it will get into portal system, hypophyseal portal system, and it will take the peptide, that tiny peptide from the hypothalamic neurons to the anterior pituitary gland, and in the anterior pituitary gland it will act on the what kind of cells? ACTH cells. What kind of cells? ACTH cells. So, answer my question, in the ACTH cells will you get receptor for CRH? You will. Okay, you have them. Okay. So, under the influence, okay, now in the background of what we discussed shortly in last couple of minutes, can you interpret for me what is the meaning of that experiment? Mice with what? Mice with homozygous disruption of corticotropin-releasing hormone gene. Means what is it? Both. You see, there are two copies. Are you with me? Yes or no? Homozygous, both the copies are what? So, that this particular, in that particular mouse, both the copies of what gene? No. CRH gene, okay, is non-functional. As a result of that, that mouse, okay, is not able to generate CRH. As a result of that, it is not able to stimulate what? ACTH. As a result of that, it is not able to release what? Cortisol. As a result of that, what effect are you getting? Can you interpret for me the latter part of the sentence? There are no, which means die at birth due to what? Pulmonary. Which means what? There aren't, isn't. Have you followed the entire story? Means this. On what basis we are saying that cortisol doesn't, on the basis of such experiments, okay, we come to the conclusion that cortisol has an important role in the maturation of the alveoli of the. Second, yet another very important role that is played by cortisol cortisol is often called as a stress hormone. Remember this, okay, is a stress hormone. Okay. Now, what is this stress hormone? Well, stress is a part of normal life. Okay. Can we imagine life without stress? Hello? It would be great, wouldn't it? Okay. But no, that does not work. Okay. So, actually people say that little amount of stress is also good, keeps us fit, you know. Okay. So let us forget about humans. Okay, and let us talk about the animal world. So, what are the different kinds? The author has given us a list. Trauma of almost any type, wound, injury, whatever, accident, infection, intense heat or cold. Just go through the injection of epinephrine, surgery, injection of necrotizing substances like an acid, restraining the animal. Okay, even if you put a mouse in a cage, a tiny cage, so that the guy does not have enough room to move around. Imagine if I put you in a cage like that, how will you or I feel? Okay, that is a stress. What is it? What are we doing right now? What are we doing? We are just trying to enlist the different factors which can cause stress. Okay, and if I were to extend this list to the humans, okay, I can add to the psychological stress. Okay, examination can be a stress, not getting success in the stress, failure in the stress, rejection. I mean, they are all different forms of stress through which we all, at some stage in life, we have gone through or will go through. So, these are all any debilitating disease. It has been found that in all these cases, stress of any kind, the cortisol goes up. And this was discovered by, he was a Canadian scientist. And very early, I think almost 1935 and 40, he made a very interesting observation that in the animal, in the mouse that was subjected to any kind of stress, different kind. In the previous one, that's why I gave a list of different kind. You see, no matter what kind of stress, he found very interesting observation. He found that in the adrenal cortex of that animal was always overgrown. Do you appreciate the observation? His observation, I am talking a very old thing. I mean, his observation is of 1935-40 or so. He found that adrenal always, no matter what stress you give, we had a long list in the previous one. Adrenal was always large, and then he found that there is a involution of the thymus. Thymus was always low, and the mass of the lymphoid tissue, lymph glands. We talked of lymph system and lymph gland, and that was always suppressed. What are we talking about? He is talking about general observation that whenever there is a stress, no matter what is the reason behind the stress, you consistently get some changes. And what were the changes? Number one, adrenal cortex was always large as compared to the normal or the unstressed animal. Thymus was always reduced in size, and the lymphoid tissue was always also reduced. Got it? This he called as a stress response, and later on it was discovered, much later we discovered a whole range of changes which I will talk about shortly, including this slide.